Hi, I'm Bob. I'm your virtual lab assistant, and this is Peter Doherty Building, where we are located. Please follow me to level 5. This is Rogerson group location. This is our office area. Rogerson's laboratory studies the pathogenesis and immunology of infections with malaria parasite Plasmodium falciparum in humans. The laboratory studies are linked to field studies, and they collaborate with leading malaria groups in Africa, Asia, and Papua New Guinea. Malaria is a life-threatening disease caused by a Plasmodium parasite transmitted by the bite of infected mosquitoes. Malaria is predominantly found in tropical and subtropical areas of Africa, South America, and Asia. In areas with high transmission, the most vulnerable groups are young children who have not developed immunity to malaria yet, and pregnant women whose immunity has been decreased by pregnancy, especially during the first and second pregnancies.
Now let's head to the lab.
This is our laboratory area. Please wear your PPE as you enter the lab. This is our culture lab, where we culture Plasmodium parasites and we can look what they look like under the microscope. We also culture different cell lines in here, or can work with primary cells. Now, let's meet the people.
Hi, I'm Stephen Rogerson. I'm a professor in the Department of Medicine. I'm head of the Malaria Laboratory here at the Doherty Institute, where we work on the immune responses and pathogenesis of malaria infection, particularly Plasmodium falciparum infection, and how it affects pregnant women and young children. We're using a variety of culture techniques and immunological techniques to try to understand and identify immune responses that may protect against the circulating malaria-infected red blood cells, and then using machine learning techniques to try to analyse that immunological data to try and unravel which are the key targets of immunity to blood stage malaria and which are the key immune responses which help protect against infection, with the idea of trying to identify particular targets that might form malaria vaccines in the future. So we're using a bunch of different types of laboratory techniques, from flow cytometry to culture to ELISAs, and putting all that data together in complex statistical analysis with our statistical partners to try and identify the main targets of immune response to malaria. I hope we'll see you here to join us in this quest to try and identify the best approaches to curing malaria and preventing it in pregnant women and young children. Thanks.
Hello, my name is Siddharth Mahanti. I'm an associate professor in the Department of Medicine and an investigator in Stephen Rogerson's malaria group. I have two projects in which students are involved. One relates to malaria immunology, and the second project is on SARS-CoV-2, the cause of COVID-19. The malaria project relates to a type of immunity called transmission blocking immunity, which is immunity directed against sexual stages of malaria that are taken up by the mosquito. The particular stage I'm interested in is called a gametocyte, and we're trying to identify antigens that are expressed in gametocytes that could be targets for preventing transmission to the mosquito and to other humans. The SARS-CoV-2 project is a study that I'm leading that's aimed at identifying factors that increase the risk of transmission to healthcare workers in hospital settings. We are also in that process trying to understand the early immune response to infection in these healthcare workers that get infected. The laboratory is a very collegial place, and the students have a very good experience both in learning new techniques of immunology as well as in learning the process of research and the types of experience that research can lead to. I hope you get a chance to come and see the labs and to see the type of work that's going on here. Thank you.
Hi, My name's Liz Aiken and I'm a researcher here at the Department of Medicine at the Doherty Institute of Infection and Immunity. The research I'm really interested in is trying to identify what kind of antibody responses protect women from malaria. That might seem very specific. Why are we so interested in pregnant women? But the problem is pregnant women are much more susceptible to disease than their non-pregnant peers. So what we do is we are interested in measuring lots of different antibody responses, and we do this because the problem is we don't know exactly what kind of an immune response a pregnant woman needs in order to be protected from disease. So we measure a huge variety of responses and we then identify which ones are most or best associated with protection. This kind of approach is can be called system serology. So the honours project that we're really interested in doing is we want to measure a specific antibody response in a cohort of pregnant women from a malaria endemic area. Then we're going to look to see whether this specific antibody response is associated with protection from disease. So for example, if these women has this specific kind of antibody response, are they less likely to get malaria in pregnancy? Are their children less likely to be born with a low birth weight? This kind of information is really important for the validation of immune responses in vaccines. So if you're interested in this kind of work, look us up on the Peter Doherty website and contact us by email. We'd really like to hear from you. Thanks a lot.
Hi, my name is Masarat Kasa. I am third year PhD student at Department of Medicine, Peter Doherty Institute, University of Melbourne. I study antibody response to malaria infection, specifically in pregnant women. I do a lot of experiments and ratings as well. So my typical day in lab is first I come and plan for experiment, write protocol, and do experiments. And then after that, analyze the data and then more readings for the next experiments. I am very lucky to work in such amazing environment in which everybody wants to help. There is a very good team spirit. We have also, apart from lab, we have also some social gatherings, including family dinner, having coffee and tea together. And I think you will love it. So please come and join us.
Hi, I'm Isabel. I'm a second year PhD student in the Rogerson lab. My project focuses on naturally acquired immunity to malaria in young children. And I really like this project and the other research that we do in the lab because we have the opportunity to relate what we observe at the molecular level to the clinical level. So we're very fortunate to work with large clinical sample sets. In my case, they come from Papua New Guinea and Malawi, these malaria endemic areas. I also really like our laboratory group because all the supervisors and students are very supportive and encouraging, not only with our professional development but also with our personal development. I think it's a really positive environment to study and learn. Hope we can see you soon.
Hello, I'm Dilini. I'm from the Rogerson lab. So I started working here as a PhD student in 2019, and since then I've been working with neutrophils and complement and antibody responses. So it's if you come here, you will see that it's an international lab. We get a lot of international students here, and it's a very friendly lab, and we are like a part of a family. I invite you all to join with us, so to be a part of this amazing family.